the cleavage when it is developing, what happens is slowly there will be a furrow formation. The cell will undergo a slight depression, and during that depression, the depression will be going deeper and deeper inside, and it will lead into a small constriction, going penetrating deeper inside towards the downward from one axis to the other axis of the egg. What is this one axis and the two axis of the egg? One is an animal pole, the other one is a vegetal. What do you mean by animal pole? Animal pole will contain large number of cells that are there, and vegetal pole will consist the yolk. Now, if you have observed the egg, you will see that towards one corner there will be large amount of the protein, and towards one corner you will see the yolk, isn't it? So in that is the place where yolk material is present large, that place is called as the vegetal pole, because later on it will be supplying the vegetation to the developing embryo. That is the reason why it consists of the nutritive material. It is called as a vegetable pole. And the pole where the cells are present and the division of the cells are taking place, that is called as the animal pole. So cleavage is initiated by the appearance of a small groove or constriction called as cleavage furrow. This furrow appears first at a point of an egg. In Amphioxus, the first furrow appears at the animal pole. The furrow deepens and extends downwards to on both the sides. That is not on one side. Just like apple. When you're taking an apple, you're giving only superficial cut, then it will not divide into two. So from the top of the apple till bottom, you will be taking the cleavage. That furrow will go from one side of the cell to the other side of the cell. And once it deepens, it will give rise to two different cells. So this furrow deepens continuously and extend downwards on both the sides. And they meet at the vegetal pole. The two ends meet at the vegetal pole, wherein that is, it starts with the animal pole and then meet at the vegetal pole, thereby dividing it into complete two cells. The furrow then extends inwards radially, finally constricting the egg into two blastomeres. The cleavage furrow divide the egg at different angles and plane. So generally the constriction starts, it starts dividing. Now, coming to the solid ball of cells, based on the vegetal pole and based on the animal pole, we know that the cleavage cannot occur in the vegetal pole. When it cannot occur in the vegetal pole, how the cleavage is occurring? Now, how the plane is going from animal pole to the vegetal pole. So based on those, we will see that there are four types of the planes. One is meridional plane, vertical plane, equatorial plane, and latitudinal plane. I will just explain this in terms of a fruit. When you take an apple, the seed is very small. So what will you do? You will cut right from the center. You start at the beginning and then you will go up till the bottom. Divide it into two equal halves. That is meridional plane. Coming to vertical plane, it is also same. Starts from the animal pole, goes to the vegetal pole, but it will not be in the center. It is not in the center of the pole. Then coming to the next one, that is equatorial plane. Equatorial plane, again, suppose if you want to cut a watermelon, how will you cut the watermelon? Not longitudinally. Horizontally, you will cut two equal parts. So same way, the division that is dividing the cell at the equatorial plane or the middle section, it is called as the equatorial plane. Whereas latitudinal plane, I gave you an example of a mango with a seed. You will, if you want to only the seed not to be cut, you will chop off the upper part. So that is any cleavage that is occurring in the midline, or that is horizontally, but not from the center. They are called as latitudinal plane. Let us go into little details. Meridional plane, the cleavage furrow bisects both the poles. Start from this pole to the other pole, passing through the polar axis. This is animal pole, vegetal pole. This is the egg center. From there is a division that is occurring. This is dividing it into two equal halves longitudinally. This is called as a meridional plane. I think you know about latitudes and longitudes. The main longitude is called as a meridian. So the division that is occurring longitudinally, it is called as a meridional plane. If you see the horizontal, this is the X center. This is the meridional plane. Either this way or this way, it is a meridional plane. And on the sides, you can see that it is a vertical plane, not from the center. If they are coming on the sides, this is called as a vertical plane. So vertical plane resembles the meridional plane because the furrow tends to pass the animal pole to vegetal pole, but does not pass through the median axis. Does not pass through the median axis. It appears on one side of the axis. Coming to equatorial plane. In equatorial plane, it bisects the egg at right angles to the meridian. This is one thing, right angle. You can see this. This is longitudinal and this is horizontal. So it is perpendicular to the first axis. As a result, we get four cells. If it is at the equatorial, both the cells will be equal half. If it is not at the equatorial plane, then the upper cells will be smaller, the below cells will be longer, giving rise to micromeres and macromeres. Did you understand this? So, equatorial plane, it bisects the egg at right angles to the median axis, halfway between the animal pole and the vegetal pole. Now, this you can see it in case of sea urchin. Latitudinal plane. It cuts the egg at right angles to the median axis as the equatorial plane, but it passes either above or below the equator of the egg. So, depending upon these planes, we get different types of cells, whether it is a equal cells or it is non-equal cells, etc. Based on the type of the egg, we get different types of cleavages. What are they? They are holoblastic and meroblastic. Now you might get confused. What are these cleavages and what are these? Holoblastic and meroblastic, it depends upon the nature of the yolk. If yolk is very less, that is in a microlecytal egg, then complete cleavage can occur. Whereas when the yolk pattern is more, it is called as a meroblastic, where only the superficial cells will be dividing and a yolk will be providing the nutrition. In the following subsequent lectures, I will be showing the pictures of it and then you will be able to understand. Now, whereas the planes, when we are talking like how it is been, whether it is longitudinal division occurring or it is the horizontal division occurring, or if it is occurring horizontally, is it at the equator or is it only on the top or is it only at the bottom level with dividing into micromeres and macromeres. Whereas if it is occurring from longitudinally, is it from the center or it is giving rise to two different types of the cell, small cell and a big cell. So based on that, that is the plane. Whereas based on the type of the yolk and the nature of the egg, we have two types of cleavages, that is holoblastic and meroblastic cleavage. Coming to holoblastic cleavage. Holoblastic cleavage, it is in this particular cleavage, the entire egg will be dividing. That is, amount of yolk is not influencing it and it is not restricting it. Now, even if you see the boiled egg, when you are cutting it with a knife, the white portion will cut very softly, whereas the yolk portion, little pressure you have to apply because protein is quite thick and it requires little more. So even there, the yolk will restrict the cleavage pattern. So that is how we get different types of cleavage that we will be talking at the end. It is total or complete cleavage. Holoblastic cleavage is called as a total or complete cleavage. When blastomeres are equal, like here, this is a zygote where it has undergone the cleavage. Both the cells are equal. It is called as a equal holoblastic cleavage. This is the cell where you are seeing that one is small, one is big. So holoblastic unequal cleavage. And this holoblastic cleavage can be of depending upon the nature of the cells that are being produced and the symmetry that you see in the cleavage. It leads into 4 or 5 types. They are bilateral, radial, bilateral, spiral, biradial, and rotational cleavage. Radial cleavage. Now, this radial cleavage, when you see, now, blastomeres are arranged radially. Radially, in a sense, what do you mean by radial? Do you remember wheel? From the center, the spokes of the wheel come out, radiate out. That is called radial arrangement. So blastomeres will be arranged radially around the central axis of the egg. The embryo has a symmetry of a wheel. Just imagination of a wheel. From the center, the cells will be moving towards the outer. The arrangement of the cells directed towards the outer side. So that is called as a radial symmetry. Any plane passing through the major embryonic axis divide the embryo into symmetrical halves. The embryo has radial symmetry. When any plane is passing through this particular axis and dividing it into two similar halves, it is called as a radial symmetry. So when the cells are arranged in a radial fashion and the symmetry, now what it is exhibiting when the division is occurring is a now both the cells equal, then it is called as a radial symmetry. The cleavage producing symmetrical embryo is called as a radial cleavage. Example, you will see it in sea urchin and synapta. Let us see. In case of one paper is missing here. Now, my daughter has missed one paper. I'll just read, explain it to you. It will be easy for you. We missed to take one more thing. Sorry for that inconvenience. When you see this radial cleavage, in case of synapta, uh, when you see in case of synapta and C. cucumber, this radial cleavage that is there, the first cleavage that is when you see the uh, like synapta or the sea cucumber, that is sea urchin, when you see this, the first cleavage that occurs, it will it will be like vertical. It is a vertical or it is the meridional cleavage that occurs. Then this meridional cleavage will occur first meridionally and at right angles to the other one. Just like you are cutting apple into four pieces. First what meridional, second meridional. That is vertical cleavages, two vertical cleavages at 90 degrees each other. Are you imagining?
Yes, yes, ma'am.
Yeah, it is two vertical cleavages which is dividing one apple into four equal parts, vertical cleavages. But each of the division is at 90 degrees to each other. Then next cleavage, that is the third cleavage that is there, it is equatorial cleavage. And this equatorial cleavage is at right angle to the polar axis. What do you mean by right angle to the polar axis? That is in the center. Suppose there are 8 children. Mother doesn't want to give 8 equal longitudinal sections to the child. She wants to give make it 4 and then divide it into 8 by putting central division. At the time, you will get 8 equal portions, equatorial position. So first two are vertical at right angles to each other. Then the other angle is perpendicular again. It is perpendicular to the vertical axis. Perpendicular to the vertical axis will be equatorial division. So dividing it into 8 equal type of cells. So these are produced in equal cells. The number of cells that are being produced, they are equal in size. So 8 different cells. So how these cells are being arranged? These cells are being arranged one above the other. So one tier of cells, two tier of cells, three tier of cells. Just imagine one apple, two vertical divisions, and from the central. How are they arranged? They are being arranged one above the other. Now next, what happened? The next cleavage again, it will be a horizontal cleavage, which is dividing them into again the different type of cell. That is horizontal. Third and fourth and fifth cleavage, again it will be horizontal cleavage. It will give rise to the 16 cells or the 32 cells. Once it gains into the 32 cell stage, then the cleavage becomes irregular. Sometimes it will be horizontal, sometimes it will be equatorial, wherein large number of cells are being divided. So till 32 cell stage, we will see that the divisions are in a particular plane. Whereas in case of sea urchin, when you see in case of sea urchin, Amphioxus and frog, you will see that the third cleavage, it is not at the equatorial region. It is above or below the equatorial region, which gives rise to micromeres and macromeres. Did you understand? I repeat this part once again. Radial cleavage, it is the cleavage which is passing or dividing the egg into two symmetrical halves where the cells are radially arranged. The arrangement of the cells in the morula is radially, and it gives rise to the radial symmetry when the horizontal cleavage is been given. Now, example is sea urchin synapta. In case of synapta and sea cucumber, you see that the first two planes are meridional, that is vertical, and that is from the central it is passing, giving rise to 4 equal cells. Then the third division is equatorial division, which will divide it into 8 equal halves. And then again, there will be a 16 cell stage where it will be dividing the cells into the longitudinal division. That again, 2 longitudinal divisions will be there, and then the 2 equatorial divisions will be there, giving rise to 16 cells, 32 cells, etc. So, whereas in case of sea urchin, Amphioxus and frog, you see that these cells, they are the third division that is occurring. The third division is not equal division. It is not at the equatorial plane. It is either above or below the equatorial plane, giving rise to micromeres and macromeres. When this type of cleavage is occurring, it leads to the formation of the blastocyst stage of cell. Then next one is the bilateral cleavage. Here I missed one paper, so I'm very sorry about it. Next cleavage is, in the next class, I will be focusing on those. I will just share those PPTs also. Bilateral cleavage. Now, this bilateral cleavage, that is, the cells are arranged bilaterally. That means when you pass on the section, you will get two similar mirror images. And when the division is occurring, you will see that the first cleavage that passed whichever plane it is passing, the second cleavage will be the horizon. That is the second cleavage, that is, it is perpendicular to it. And this will give rise to the anterior and the posterior side and right and left side of the embryo. In case of bilateral symmetry, you will see that the meridional cleavages will be there, the plane of cleavage that is there, the second cleavage will be perpendicular to it, and the cells that are being formed, they are completely the mirror images of the each side. So here, when the division occurs, we have a right side of the cells and the left side of the cells, and it will be giving rise to the polar axis, anterior and posterior polar axis. So such type of symmetry, which are giving rise to two lateral halves, it is called as a bilateral symmetry. Generally, this you will see it in ascidian. Ascidian is, have you heard about Herdmania? In an ascidian, you will find this type of cleavage, that is a bilateral cleavage. Now, during this, you will be able to see that during the bilateral symmetry, the 4 cell stage, when you see the cells will be arranged, or it appears to be 4 different blastomeres. They are all dissimilar blastomeres. Though we have a right side and the left side of it, but still you will see that the cell size will be dissimilar. All the cells will be dissimilarly arranged. They are of not equal size. The third one is the spiral cleavage. Spiral cleavage, I will take it in depth because that is an important question either in your CSIR or NET exams, the spiral cleavage, or even in your examination also, they will ask you about the spiral cleavage because it is a unique feature. And because of the spiral cleavage in molluscans, you will be able to see a different type of symmetry. So I will take up spiral cleavage in detail tomorrow. Then biradial cleavage. The fourth type of cleavage is biradial type of cleavage. Here, this occurs in ctenophora. Ctenophora. What is ctenophora? It is a, what is ctenophora? Minor phyla. What do you mean by minor phyla? Any idea? Major phyla and minor phyla? Anybody? Do you have any idea about major phyla and the minor phyla? Sleeping?
No, ma'am.
Hmm. Major phyla are all your the invertebrate and vertebrate phyla, all the protozoa, porifera, etc. You know that they have a defined characteristics. If I say protozoa, you will remember all the characters of a protozoa. Any animal, if I take protozoan animal name, you will be able to identify the character. Whereas these ctenophorans, they have the affinities of both two different phyla. They will have it is not a connecting link as such, but they have their own unique characteristics and they have some similarities with the preceding phyla and the descendant phyla. So both the phyla they have but they have their own unique characteristics. They have their own unique characteristics, but apart from that, they also have certain characteristics of the preceding phyla and the succeeding phyla. So such type of phyla are called as a minor phyla. They cannot be categorized in the major phyla. They are totally a separate phyla itself. Whereas a connecting link means we will be able to know the characteristics. The major characteristics will be of one particular phyla and they have the similarities. Like peripatus, we have studied. Peripatus, it is an arthropod, isn't it? Because it looks like an arthropod and it has a jointed legs, etc. But it has annelidan characters too, affinities to annelidan character. Therefore, it is a connecting link. Whereas in Ctenophora, you will see that it has its own unique features. It has its own unique feature, but it appears to have certain similar characteristics with the phyla which is preceding it and it is succeeding it. So such type of phyla, minor phyla, in such you will see the biradial cleavage. In biradial cleavage, you'll see that first two cleavages are meridional, third one is vertical. All these what we were seeing, the third was horizontal. Whereas here the third is vertical again. That is two meridional will be there and the third will be there. Therefore, in this biradial, you will see one larger group of cells and a smaller group of cells. So here you will find a curved plate of cell arrangement because you just see, imagine when the apple is being cut from two angles, that is one meridian, then again another four parts it has been cut. Next, instead of cutting it from the center, you are cutting it again towards one corner, that too vertically. So what will happen? So one part of the half of the apple will be again cut into smaller pieces. So you will get a disc pattern of the cell arrangement. So they are being arranged in two rows of four each, and the central cells will be longer than the other cells. So here in biradial, you will have two different type of cell sizes because the third cleavage happens to be vertical cleavage. So in this, when you see that the third cleavage is vertical, you will not be able to see the similar structures, whereas you will see the curved nature of the cell, where discoidal structures of the cell arrangement can be seen. Coming to the next one, that is a rotational cleavage. This is generally an unusual cleavage which we come across in mammals. Here, the first cleavage is meridional. The first cleavage is meridional, just like all the other animals. It produces two blastomeres. And the second cleavage, it is unique again, resulting in unusual arrangement of blastomeres. Why? Because what happens here is the plane of the second cleavage, it will be changing. So in the second cleavage, what happens? One blastomere in the plane of the cleavage passes through the polar axis. But the second blastomere that is present, that plane will be perpendicular to the polar axis. So in rotational cleavage, the first one is a meridional. Then this second cleavage, what happens? The cell will be rotating. When the cell is been rotating, you will not get an equal. One axis, one will be polar, the other one will be in a other direction. So one cell will be cut in one direction, the other cell will be cut in a another direction. I will give you the example of this rotational cleavage. Here, what happens is, if you are taking an apple, if an apple is solid and fresh, you can give equal cut. But if apple has been kept for longer time in refrigerator, what will happen to the apple when you take it out? It becomes quite soft tissue. Now, during that apple, if you cut, you will not get two equal halves. First cut, second cut. You just imagine you will get four different types of cells. And then when you are cutting it again vertically, you will not get equal size. One will be thin, one will be little stouter. So similar type of divisions will be occurring now in the blastula, in the zygotic stage, wherein first cleavage that has occurred, that cleavage will divide it into two. The second cleavage will also be horizontal. That is, it should be at 90 degrees to the first cleavage. During that time, what happens? There is a 90 degrees rotation of these cells. So during that 90 degrees rotation, one cell will have a cleavage in one angle, the other cell will have a cleavage in another angle. As a result, what happened? You will see that the perpendicular, that is the cell, each division, it will be a perpendicular division at 90 degrees. So blastomeres will be rotating at 90 degrees, and therefore each cell will be cut at a different level. And this cleavage continues, and it gives rise to uneven number of cells, and it has a various type or various structure of cell. But this cannot occur in human beings, because if it is unequal cell stages, say for example, when this blastula is converting into a gastrula, where the fate maps we are talking about, if the number of cells for forming a nervous system is very less, and the nervous system will be a deteriorated nervous system, so such kind of cleavages cannot occur in human beings. In mammalians, it's a condition where the cells undergo the rotational change, where generally where this differentiation is not occurring. In those cases, it will be occurring where the cells will undergo 90 degrees rotation after every cleavage, and therefore there will be unequal divisions occurring and giving rise to unequal blastomeres. And then comes the spiral cleavage. I will take up the spiral cleavage in the next class. And the next type of cleavage is meroblastic cleavage. Meroblastic cleavage, again, it is divided into discoidal and superficial cleavage. Depending upon the potentiality of the cells that are there, you have a determinate cleavage and indeterminate cleavage. Then I close my lecture with factors affecting the cleavage pattern. So for today, I will stop here. I will go for the spiral cleavage in the next class, because for spiral cleavage, you should be able to understand. You have just, it's very difficult to understand the rotational cleavage itself. For spiral cleavage, it is more difficult. Why? Because we have to talk about the spindle axis. So here, when a division is occurring, one cell, the division will occur at the equatorial plate. The spindle will be formed in the equatorial region. For other cell, the spindle will be forming in the other direction. So therefore, you will get a different type of the cell structure. That with the diagrams, I need to show it to you. That's why I'll take the spiral cleavage in the next class. Is it okay for today?
Okay,
Tomorrow,
ma'am.
Okay,
tomorrow
ma'am.
I will take up the spiral cleavage and the remaining part of the cleavage, and gastrulation also I will finish it off.
Okay.
So tomorrow's portion, both spiral cleavage and gastrulation will be done.
3 o'clock, ma'am.
Tomorrow 3:15. My scheduled time is at 3:15 only. No, that link will open only at 3:15.
Fine, fine.
Okay.
Thank you, ma'am.
Did you understand?
Yeah, ma'am. Little bit, but we need to do a homework also, no, ma'am.
Yeah. No. Now, what is the difficulty?
What type
Terminology
of
or
questions
that?
will come in
Yes,
this
ma'am.
topic, ma'am?
Yes,
Ma'am,
ma'am.
what type of questions it will come in this topic, ma'am?
No. Once I complete the complete topic, I will tell you.
Okay.
This is a favorite topic even in your entrance examinations, as well as your generally, what is cleavage? How many types of cleavage do you find? That is a major question they ask.
Okay.
This, it is very easy if you understand.
Yeah, ma'am.
What was it? Yes, ma'am?
Nothing, ma'am. I
No,
said
somebody
okay.
was somebody was asking something.
Okay.
No, no, no. Nobody is something asked.
Okay, fine. So if it's a language problem or the speed problem or the topic means new terminologies and all that. Yes,
But in this it will come major question, ma'am. This cleavage
major
is
question.
major.
This is definitely will be a major question.
Okay.
It's a favorite question. Major question means it's a favorite. Any of the examinations you see, cleavage definitely they will ask.
Okay.
Okay, fine.
And
Today Radhika is not there. I think your classmate. So who is taking the screenshot of the attendance?
should I take, ma'am?
Yeah, anybody, and post it on to my number. I think I've already given my number, no?
No, ma'am. Can you repeat it again? I'll note it down.
934
Okay.
888
Okay, ma'am.
4299.
Thank you, ma'am.
Okay, just post it to me.
Fine, ma'am.
Okay, fine. Any problem, you let me know. In the next classes, I can just improve it what it is.
Nothing, ma'am. You're telling clearly. It's a point to point.
Okay, fine. Thank you. Okay, shall I call it a day today then?
Sure, ma'am.
Good evening. Okay, bye.
Yeah.
हेलो मम्मी. हाँ मम्मी, क्लास ले रही थी मैं. क्लास ले रही थी मम्मी. क्लास ले रही थी मम्मी मैं. घर में ही हूँ. कॉलेज जाके माँ. पता नहीं मम्मी. हम्म. ठीक है. रखती हूँ. बाय.